In this slide set, we will review the structures and functions of nucleic acids, including DNA and RNA, and their roles within cells. First, we'll start with a quick review of both the molecular components of DNA and RNA. If you recall, the molecular components of DNA and RNA are called nucleotides. And there are two different types of nucleotides, if you will. There are those that are composed of a sugar backbone that contains the sugar deoxyribose. Notice the lack of the hydroxyl group here. And those nucleotides found in DNA contain this deoxyribose sugar. Alternatively, there's those nucleotides found in RNA. Those are composed with the ribose sugar, and notice the presence of the hydroxyl group here at the 2' position. In addition to the sugar backbone, there's also a triphosphate group that's part of both the DNA and RNA nucleotides, as well as this base group. And so together the base, the sugar, and the phosphate equals a nucleotide. Nucleotides, in addition to describing the molecular components of DNA, are frequently used as units of measure to describe the length of a DNA or RNA molecule. For example, a segment of DNA or RNA can be said to be a specified number of nucleotides long. In addition, nucleotides can be a measure of distance. In this case, the nucleotide unit describes the number of intervening nucleotides between one position in a DNA or RNA molecule and another. For example, if we're studying a particular mutation, we may say the mutation in gene A that causes a particular disease is 25 nucleotides away from the start of the gene. And so this is just an example of how you may hear the different ways of the same term being used. Next, let's take a specific look at those bases that are part of the nucleotides that make up both DNA and RNA. As you see here, there are two different groupings of bases, or three I should say. There's bases that are found in both DNA and RNA. These include adenine, cytosine, and guanine. Then there's the nucleotide thymine, which is found only in DNA, and uracil, which is found only in RNA. And so the different bases or side groups attached to the amines differ among the bases. Now what you notice is, or what we will see is, in DNA we will always see the nucleotides A, C, G, and T, whereas in RNA we'll only see the nucleotides A, C, G, and U. And so in that case, anytime there's a T in the DNA, it's replaced with a U in the RNA. Now, if we take a closer look at the structure of both DNA and RNA, what you see is that the DNA and RNA nucleotides join through a single phosphate group, see that here, to form a string of DNA or RNA nucleotides, or a strand of DNA. This is what we would call a strand of DNA or a strand of RNA. When deoxyribose sugar nucleotides join, as we had said, they make a DNA strand. When ribose sugar nucleotides join, they make an RNA strand. And what you'll also notice is what we often do is, rather than draw out these very complex molecular structures, we'll simply abbreviate how the different nucleotides are put together by using the single or first letter of the different bases. And so here you see a linked combination of nucleotides containing cytosine, thymine, adenine, guanine, cytosine, thymine, adenine, guanine, etc. And you notice the simple use of the first letter, CTAG, CTAG. Now from this alone, you know that this is a DNA molecule. And you might say, well, how do you know that? Well, if you remember, the presence of the thymine indicates that it's DNA. If instead of thymine we had uracil, or a U in here, each time there was a T instead, we would see a U, then you would know that that's likely an RNA molecule. One of the most significant findings of Watson, Crick, Wilkins, and Franklin was that at the physiological pH and temperature of living cells, DNA usually is found as a double-stranded molecule. And when DNA forms a double-stranded molecule, the strands are joined together through hydrogen bonds, and you see those here highlighted in green. And it's these hydrogen bonds between the bases. Because of the specificity of the hydrogen bonding, A always pairs with or is complemented by T, and C always pairs with or is complemented by G. And the two strands of a double-stranded DNA molecule are said to be complementary. As shown, AT base pairs form through 2 hydrogen bonds, and CG base pairs form through 3 hydrogen bonds. And once joined together now, the bases are referred to as base pairs. And so you'll see this consistent base pairing, A always pairing with T, C always pairing with G. What this allows us is the complementarity of base pairing means that if you know the nucleotide sequence of one strand of double-stranded DNA, it allows you then to predict what the nucleotide sequence of the other strand will be. Moreover, in terms of terminology, the length of a double-stranded DNA molecule often is described by the number of base pairs in the helix. So you said, as I mentioned previously, you could count the number of nucleotides, but sometimes you also see some reference to whether a gene is 28 base pairs long, for example. And this would mean that there's 28 nucleotides lined up with their complementary nucleotides on the other side of the double-stranded DNA. Now, RNA molecules, I'll say before we move on, are a little bit different. RNA molecules within cells typically are single-stranded at normal physiological temperature and pH. However, they frequently fold up upon themselves to form some secondary structures, wherein a single RNA molecule forms intramolecular base pairs. And as with many other biological molecules, the shape created by folding affects how ultimately the molecule will function inside the cell. Now, although one RNA molecule may associate with another in some circumstances, RNA is not typically found in double-stranded structures. And so in terms of exams or generalizations, it's fine to say DNA is double-stranded and RNA tends to be single-stranded. And along with that, there's also a measure of stability. DNA being double-stranded tends to be more stable than single-stranded RNA. Now, in addition, although it is double-stranded, DNA also takes on another structure. So double-stranded DNA is not a straight ladder-like molecule, as kind of is indicated in the top of the slide here, but it instead turns in a helix like a spiral staircase, as you see here on the bottom. And the DNA helix is called a double helix because it is made of 2 complementary strands of DNA. The paired bases lie in the center of the spiral, and you see them here, the C's pairing with G's and A's and T's pairing together. Well, it's the sugar backbones, or sugar and the phosphate groups, I should say, that form the backbone of the DNA double helix. The hydrogen bonds between the base pairs hold the 2 sides of the helix together. But I also mentioned that these hydrogen bonds can be broken fairly easily. And the DNA double helix can be forced to separate into the 2 component strands if you increase the temperature or the pH of the solution in which it is suspended. And if the temperature and or pH is lowered back down again, interestingly, the complementary DNA strands will find one another in solution and sort of rehybridize or bind together again into the double helix, in which A's again will pair with T and C's will pair with G for the entire length of the strands. And it is these properties, these physiological properties of DNA that we can exploit in the lab to be able to study genetics better. But the body also, or the cell also, exploits this physiological property. And so in order to replicate the DNA, the DNA molecule will have to separate or be broken apart, as well as for transcription. And now replication is the process by which a DNA molecule reproduces itself. Every time a cell divides, it needs to replicate its DNA. And transcription is the process through which DNA serves as the template for the synthesis of RNA. And so in order to get RNA to make protein, and we'll talk about that in a bit, the DNA again needs to be separated so that an RNA copy can be made of the DNA. Now, before we go any further, sometimes there's a little confusion about the hierarchy of what we're discussing. And so if we start out with the basic functional unit, and that being nucleotides, you see here in this image the nucleotides being A's, T's, C's, and G's. From there, this is what I have in the image as an example of a genomic sequence from a virus that we had worked with in the phage explorations course here at UAB. And so, but this could essentially, you'll see the same thing in the sequence of any type of genome, just lots of different letters all lined up. And this is how the different nucleotides are arranged together in this particular genome. The nucleotides then are combined together to ultimately form a strand of DNA, as we had said. And here you see the double-stranded helix, or the double-stranded DNA forming the helix. And then that is then ultimately in humans organized into more complex structures known as chromosomes. And so the DNA from each parent are arranged into chromosomes. There's 23 chromosomes from each parent, 22 pairs of autosomes. These are just regular general body chromosomes, if you will, and 1 pair of sex chromosomes. So ultimately, I should have written 23 chromosomes from parent equals 46 chromosomes. There's 46 chromosomes in the human body, 22 pairs of the autosomes, and then 1 sex chromosome from each parent. And so what you see on the figure on the right here is just a basic description from what we know today from the Human Genome Sequencing Project of the 22 chromosomes, how many genes have been located or identified on each one of those chromosomes, how many bases there are, and this is essentially, again, a count of the nucleotides. And so you see in the human genome, just extraordinary numbers of bases make up our particular genomic structure. Now, let's move on from there to discuss DNA replication. And so there's two different main time points in which DNA replication will occur: prior to mitosis, and this is the process when non-reproductive or somatic cells divide, and we've discussed the different processes of mitosis, and also for meiosis, and meiosis is the process by which germ cell precursors divide to form the sperm, egg, and other reproductive cells. Before this cellular replication occurs, each DNA double helix within a cell is duplicated. This process, called DNA replication, begins at precise locations encoded within DNA molecules, and these sites are called the origins of replication. And so replication cannot start anywhere along the DNA strands. There are nucleotide sequences that identify where replication will occur. And then from there, there's a number of different proteins and enzymes that work together to cooperate and efficiently replicate the DNA. First, enzymes called helicases will come in and facilitate unwinding of the DNA double helix. And then once unwound, the DNA strands are replicated. The enzyme, as we had said, that's responsible for this replication is called DNA polymerase. And so DNA polymerase enzymes copy each strand of the DNA, relying on the complementarity of DNA base pairing to replicate each strand faithfully. So in essence, it's like a copy machine. The DNA helicase will come in and unwind the DNA, start to pull it apart. The DNA polymerase will sit down. That's like the copy machine. And it'll look at the two different strands, and again, they're complementary to one another, and it'll essentially make a copy of that. So it'll take the template which was the original strand, and make a new strand that matches with it. Because of the precise process by which each strand of DNA is used as the template to produce another strand, DNA replication is said to be semi-conservative. Semi-conservative means that each new DNA double helix is made up of one of the old strands and one of the new strands. And so you'll see this will separate, and these will be the templates on either side, and the new strand will be made that's complementary to the old strand. On the leading strand, the DNA polymerase is DNA, I should say, is replicated in long 5' to 3' segments. And so DNA polymerase can only operate in a certain direction on the template strand, which allows for one side of the DNA template to be made or made continuously, whereas the other side in the lagging strand is made in short fragments called Okazaki fragments. And then these fragments are then joined together by another enzyme called DNA ligase to complete the replication of the lagging strand. Now, DNA polymerase is a very robust enzyme. As you can imagine, in order to maintain the integrity of the human genome, it needs to be pretty precise in terms of not making errors when copying the particular template strands. And DNA polymerase has a built-in proofreading capability that's an error rate believed to be less than 1 base pair per billion. This is really a phenomenal number when you think about it, that over 1 billion bases can be copied, and there's really less than 1 base pair error per billion. So let's take a minute to review some of what we've learned. I'll have included 2 animations. The first we'll see from McGraw-Hill. The second is a nice Howard Hughes animation that shows shows the process of replication. So the same process just being shown 2 different ways.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the 2 complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, 2 different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Before we leave this topic, I also just wanted to quickly show this image of DNA replication because I think it shows a little bit nicer what we're discussing. Here you see the DNA double-stranded helix. Here's the DNA helicase that's coming in and unwinding that DNA and separating the two different strands. And the two different strands then act as templates for the DNA polymerase. And here on the leading strand, you see this DNA polymerase is making this brand new fragment. Here's that new fragment being made from the older fragment. And you see it, there's no breaks in the DNA polymerase on the leading strand. It can just continuously march down the template making new DNA. Whereas in the lagging strand, the DNA polymerase, because of how it has to move in a particular direction, you wind up with these different, what we call Okazaki fragments. And it's the responsibility of the DNA ligase to come in then and ligate or repair these different fragments together. From this drawing, what I really want you to be familiar with is that DNA replication is a process in which DNA is copied from the parental template strand. In essence, it's a semi-conservative replication process in which the two template strands are ultimately matched up with two newly produced DNA to ultimately produce two strands of the DNA, or two copies, I should say, of the original DNA. You should know that helicase is the enzyme ASE, indicating an enzyme responsible for unwinding and breaking apart those parent DNA strands. That DNA polymerase is the enzyme, again ASE, responsible for DNA replication. So where are the genes in all of this? When you look at all these letters, you just start to see a lot of letters. And how do we make sense of or get from these nucleotides arranged in a particular fashion to where we're actually talking about the genes? And genes make proteins, and proteins are essentially the different combinations of proteins are what give a cell a particular function. Well, the genes are in here in all of these letters, and so it's sort of like unlocking the code, if you will. Part of sequencing the Human Genome Project, the cheap part actually is just getting all of these letters. The expensive part is trying to figure out what all these letters mean, and it takes just tons of hours and human time to actually work through all of this. There are plenty of computer algorithms that have been written. They continue to get better all the time that allow us to predict where genes may or may not be amongst all of these letters. There's a few simple clues that we can rely on. Genes are indicated by start codons, so anytime you see the ATG nucleotide combination, that may indicate the start of a gene, although it doesn't always necessarily. Also, stop codons. This is where, in essence, the gene ends. There's 3 different ones that eukaryotic genes use to indicate a stop. So genes hold the code for a specific protein, and by using these computer algorithms and by human eyes, to date, there's been a number of different chromosomes we already knew were there, a number of different genes identified for each of the different chromosomes. And so here on this chart, you see the chromosome number and how many genes so far to date have been identified on that particular chromosome.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
So we talked about replication. Let's move on to the process of transcription. Now, like replication, transcription also relies on the base pairing of DNA. And again, we're using the template. In this case, though, in transcription, rather than making a DNA copy, now we're making an RNA copy. So the RNA is now being made by another enzyme. If you recall, when we made new DNA, we used an enzyme called DNA polymerase. Now when we're making RNA, we're using an enzyme called RNA polymerase. And so the process is somewhat similar. There are regions where the polymerase can bind. The RNA polymerase binds to the DNA template. Again, you have enzymes coming in and unwinding the parental DNA. And then you have the RNA polymerase binding and moving along the DNA to make a copy. In this case, it can be a couple of different types of RNA, either message RNA, this is the most common, mRNA. And message RNA is a copy, essentially an RNA copy of a DNA gene. You can also have tRNAs. These are transfer RNAs. These are the molecules used for protein production, as well as ribosomal RNAs or rRNAs. So if you recall, though, rather than making two copies of the DNA like we did when we did replication, for transcription now you only have a single-stranded RNA molecule coming off as a result of copying the DNA template. Now, what's interesting is why would we make the RNA template? And let's say in this case we're making an mRNA, a message RNA template. Well, the whole point of copying or transcription is to ultimately get from the DNA message inside your cells to what the protein is going to be. That's what a gene is. A gene ultimately indicates the sequence or the particular order of which amino acids need to be put together to make a protein. So we want to go from gene to protein. And how do we do it? Well, we first go from gene to mRNA. And so here's the mRNA molecule that's created in a eukaryotic cell. Now, all of this occurs within the nucleus, but before this mRNA molecule gets out into the cytoplasm, a few things have to happen. Our genomes are a little bit sloppy in that there contains two different types of nucleotide sequences in our genomes, or even within our genes, I should say. There's the exons, and then there's the introns. The introns are intervening, if you will, intervening nucleotide sequences that ultimately are not part of the final protein sequence. And so this is, you can think of these introns as junk. These are intervening sequences of junk. And so in order to get rid of these, the cell splices out the introns and gets rid of them to only leave the good coding exons remaining. In addition to that, there's something called a 5' methyl cap that gets put on the mRNA, as well as a 3' poly A tail. And so RNA processing in eukaryotic cells occurs to ensure the integrity of the mRNA that's going to exit the nucleus and enter into the cytoplasm and serve as a template for protein production.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
Okay, now once the mRNA has exited the nucleus, we now need to get that mRNA to become a protein. And how does that happen? If you recall back to your different cell structures, we have to think about ribosomes. If you recall, ribosomes were the site of protein production. And so this newly spliced 5' cap, 3' poly A tail, intron spliced, only exon remaining mRNA strand then exits the nucleus and binds up with a ribosome. And you see the ribosome indicated here as a yellow blob. And in essence then, it's the ribosome that reads the code of the mRNA to ultimately create the protein. Now, what is this code? Let's talk about it in greater detail. If you see here, this on the bottom here is an example sequence of mRNA. Note, there's only A's, U's, G's, and C's. There are no T's in mRNA. And so what happens is the ribosome sits down, or I should say this mRNA gets threaded into the ribosome protein. And so the ribosome looks at each of these nucleotides each triplet nucleotide, which is called a codon. So it'll look at 3 nucleotides in a row, and it usually starts out with the AUG, that's the first one, and that codes for a methionine. And so as we had said again, if you remember, ATG on DNA encoded for a start codon, the beginning of a gene. And so the complementary to that in RNA would be AUG. And so the ribosome recognizes the AUG as the start of the protein and brings in a tRNA with a methionine on it. And so here's an example down here of a tRNA molecule, some specific structural features of a tRNA. If you look at the bottom here, there's a region called an anticodon. And the anticodon of the tRNA recognizes the particular codon or nucleotide sequence in the mRNA. Now, when that matching determines ultimately which, I should say the anticodon, determines which amino acid is loaded onto the tRNA. And so when you see a UAC in the anticodon position of a tRNA, that tRNA always has a methionine amino acid tagged to it. And so in this table sort of tells us what the particular codons are. And so the codon ACU, you can look it up in the table, A in the first position, C in the second position, and U in the third position would code for threonine. And so now the next tRNA that comes into the ribosome would be carrying the amino acid threonine, and that would ultimately bind to the methionine and start to make this protein. And so the ribosome just steps down this sequence, looking at each one of these triplet codons, and then ultimately the tRNAs come in and sort of deliver, if you will. They deliver the different amino acids that ultimately make up that protein. And this is how we go from a series of letters and nucleotides in DNA to mRNA. And then the mRNA ultimately holds the different codes for which amino acids ultimately will go into the protein.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
And so let's do a quick summary of where we are right now. There's been a lot of information thrown at you pretty quickly, but in a brief summary, DNA is a complex macromolecule. It's made of several basic atomic components, and we've talked about the different nucleotides and the A's, T's, C's, and G's that make up DNA. DNA encodes genes, the instructions needed to make the mRNA and proteins. DNA is, as we said, a double helix. The complementary nature of DNA helix permits its replication as well as transcription. And we talked about DNA polymerase being responsible for the enzyme responsible for making a copy of DNA, RNA polymerase, the enzyme responsible for making an RNA copy of the DNA. And DNA ultimately, we said, is transcribed transcribed into RNA, and this is the mRNA or the message RNA. When we're talking about protein production, that's then undergoes some modifications, the mRNA prior to exiting the nucleus, and this was the 5' cap, 3' poly A tail, as well as the splicing of the introns or intervening sequences. We look at a brief summary of mRNA, or of the different RNA molecules. The mRNA stands for messenger RNA. Again, this is the template for the production of proteins, the molecules that build and comprise structures and perform enzymatic reactions. If you remember, RNA, any one of these RNAs only contain the nucleotides A, C, G, and U. You will not see a T in RNA. There's also tRNA, as I've talked about briefly. These are transfer RNA molecules. They enable the transfer of amino acids into the growing protein chain. And lastly, then there's ribosomal RNA, and these are a component of the protein-producing ribosomes. So 3 different types of RNA. The most common that's being made in the cell is mRNA, of course, because this is what the genes are using, mRNA, to ultimately make a protein. But we also see tRNA genes as well as ribosomal RNA genes.